Please welcome Markus Wanko, Institute of Science and Technology, Austria.
Exactly, that's me. Thanks very much for having me. I'm very happy to introduce Harold de Vlada and Ribbon Biolabs to this crowd here. And I'm actually introducing him in a dual capacity. Harold was both the first postdoc at our institution at ISTA, and Ribbon Biolabs is one of the first companies that we funded through our venture fund IST Cube. So ISTA, if you are familiar with it, is a relatively young research institution in Austria. The Institute of Science and Technology Austria, really across all the natural sciences and computer science. IST Cube is a venture fund that we founded a couple of years ago with private investors. And in both capacities, we're happy to introduce him. Why did we introduce him? The synthesis of long DNA in an automated and highly reliable way is obviously groundbreaking. But it's not only groundbreaking, it's also very concrete. And I think it's concrete on the demand side. I think all the other 10 companies that are here in this afternoon could be potential customers of Ribbon Biolabs. And also their production capacity is very concrete. But on this, I leave it to Harold, and he'll give you all of it.
Breaking the wall between synthetic DNA and biotech. Harold de Vlada, Ribbon Biolabs.
The computer code that generated the internet relies on 4 numbers that interconnected the whole machines across the world. That has been a leap innovation that ended up in a range of applications and products that constitute the life that we live today. In a similar manner, the DNA encoding the bioprocesses that direct life also are the base of making new and next generation of biotechnologies. Take, for example, the revolution of genomics in the 2000s and 2010s. By reading DNA strands, we could read the genome of humans and other organisms, and therefore advance a range of technologies that improved healthcare and other biotechnological areas. But an even greater potential exists in synthetic DNA, which is writing DNA molecules. This is a technology that has been ongoing for some years. But the problem is that the DNA molecules that can be synthesized are very small. For example, take the distribution of genes in the human genome. We see that the current technologies can only synthesize up to 39% of the human genomes, leaving a 61% untapped. And this is where we enter. Ribbon Biolabs is able to synthesize molecules between 5,000 and 50,000 base pairs in a seamless manner. We position ourselves as a DNA synthesis company to partner with different biopharma companies and accelerate the preclinical stages for bringing faster and more potential products to the market with our partners. We're talking about antibodies, viral vectors, gene therapies, mRNAs, hormones, and a whole range of different applications, also beyond health in life science, such as biofuels and others. For example, I'll walk you through this, which is when you see the current DNA technologies that synthesize today, the shorter the DNA is, the more molecules you can produce per run. But as the target DNA molecule is longer, it becomes harder and harder, and also much more expensive. Our technology fills the gap of what is impossible with those technologies, and we on top get the benefit of the economy of scale because we can produce at high throughput. This is, of course, something that is feeding back into the biorevolution. We are an ecosystem provider that is helping the different segments, which are the major industry drivers for the next wave of biotechnologies and health. We're talking about a market value of up to 4 trillion, according to McKinsey, in the next decade. So this is really enabling something that until today is not possible. The conventional DNA synthesis goes by putting brick by brick and synthesize a short strand of single-stranded DNA. Then you still have to synthesize the other strand to be able to make the double-stranded, the double helix that is the functional DNA. This is not what we do. What we do is we take more complex building blocks, which are short strands of short double-stranded DNAs. We combine them in pairs to make longer syllables, and again in pairs to make long DNA. If you imagine this not with 4 building blocks, but with 100 or thousands, you can make very long DNA molecules. This is essentially, we're talking about 8 to 10 base pair building blocks, which are stored in this large fridge, which is automated, and with our platform, we can synthesize up to 100 molecules per day. Ribbon Biolabs recently secured a Series A of 18 million, with which we have been developing our company and starting commercialization activities across the world. We are looking into partnering and helping the development of expression vectors for antibody therapies, for gene therapies, and many other products to help patients in need, particularly for undruggable diseases. My team is composed of about 30 people. Since I wrote this, it's outdated. We're around 30 people in Vienna and Philadelphia, and we are seeking to partner with other biotech companies, startups, or corporates to expand our team, and also preparing for a Series B investment to continue our growth. So let's partner together and write the future of technology and DNA.
Thank you very much,
Thanks,
Harold. And let's go and start for our 4-minute session Q&As. And this is a firework of Q&As. Whoa, OK, let's go.
Great time.
She was first. She was first. Then 2. Then 3.
I was
Whoa,
wondering
4.
about the cost of synthesis in your business model. I think I saw some materials referring to you actually trying to capture more of the value by synthesizing antibodies. Just curious what your plan is now.
Well, the cost of production is relatively low because we use very small aliquots. We work with microfluidics as part of our process, so we use very low amounts, so it's very cost effective. Is that what you're asking?
What the cost of a synthetic DNA would be?
The cost of the synthetic DNA, please.
Well, the cost, of course, is something confidential, but it's definitely compatible with the existing other
And then
technologies.
your business model, are you trying to co-develop, capture some of the value, or are you just selling a straight commodity?
We do not have a business model that is pay-per-base per, but in reality, value-based, together with the partners.
OK, next question is next to you. That's Frank, and then we will have Stefan.
What is the largest scale you can produce?
The scale is mostly for preclinical, so mostly for transforming some organisms. So it's not GMP grade for like kilos or grams. It's very small.
Microgram or milligram?
Yeah, that's right.
Microgram. No.
Microgram, yeah.
Okay.
OK, we see the subtleties here. Stefan, next question.
What is the error rate when you assemble the DNA?
Zero.
0.
I can explain later why.
Okay.
OK. This is the president of the jury, you know what I mean?
Well, it will take a bit longer.
It's OK, that's fine.
So, Stefan, we use pre-synthesized building blocks, which are error-proof already, and we do not use error-generative processes like PCRs. So we just assemble. So if the building blocks are accurate, our product is accurate. That's the reason, in short.
The person on the second row was the first, and then we will have this one, unless the jury has... OK, first you, but quick. Didn't you? Oh, OK, good. OK, then Frank, and then Frank, and
Well,
then
can you talk
one
about
behind. Sorry, yes.
Can you talk about the purity? Do you have also shorter products?
Well, we can make shorter products, but then...
do you have also as an impurity shorter products? You need to clean your final product?
Yeah, well, of course, yeah. In the process, you generate impurities, but at the end, of course, these are cleaned, yes.
Yeah, but what is the purity after the run?
So according to NGS, it's 100% pure, yeah. So we eliminate the building blocks that are remnant.
Great, I think you answered 2 questions at the same time. Fantastic. Next one. No, they're first. Then you are, and then you. OK, so the third row, please, the gentleman here. Yes, exactly.
How does your technology compare to established players like Twist Bioscience, for example, who can already do synthetic gene synthesis?
Yeah, so because we're streaming, I have to be politically correct. So it compares from the point of view that, I mean, what they do is great, given their technology, but they're focused mostly on oligonucleotides and gene fragments. So we start where they end. So they cannot produce anything beyond 5,000. That's where we start. So our USP is in the length and the speed of production of those long molecules.
OK, the young lady in the second row, please, is the next one, please. Thanks for your patience with me.
Since you just gave us an insight on the purity, so there's also the concept of pyrogenicity, since your building blocks might be used for cellular gene therapy.
Yep.
Do you have any insights on the pyrogenicity of the DNAs that you manufacture? Because it's a different concept than purity.
Yeah, absolutely. But that really depends on the specific use case that we're talking. So we synthesize the DNA, but actually the downstream application is with our partners. So that's beyond the scope of our product, actually.
Edgy questions. Did you already pitch here in your life? Next year, yes.
Come over.
Absolutely, and we want more women on stage, so that's great, yes. And a call to every woman here out there, you're looking at us, come on stage, we need you. But thank you very much.
Thanks to you, thanks everybody.
Yes, exactly. And we're going to take the next one.